This is Dr. Pankaj Kumar, your mentor for the session of meiosis. So first of all, we will learn about that what are the key characters of meiosis, right? So where does meiosis occur? Of course, it is a germ cells. So meiosis, as you know, was first noticed by Strasburger. Later, the term meiosis was coined by Farmer and Moore. It occurs in germ cells, right? And immature anther is the best material for the study of meiosis. Why? Because in case of immature, it is in the process of meiosis. That's why it is the best material. Okay, and anther can be easily taken in comparison to the female reproductive structure. So that's why immature anther is the best material for the study of meiosis. At the end, 4 daughter cells are formed, and all 4 of them chromosome number is reduced by half. Okay, so this is about meiosis. Now let's discuss what are the stages of meiosis. You know, meiosis can be divided into 2 parts, meiosis 1 and meiosis 2. So meiosis 1 is some sort of reductional division and meiosis 2 is the equational division. It is just like mitosis. Okay. Again, meiosis 1 can be divided into, let it be say, prophase 1, metaphase 1, anaphase 1, telophase 1, and similarly meiosis 2 can also be divided into prophase 2, metaphase 2, anaphase 2 and telophase 2, right? But out of all those things, the most important is your prophase 1, because the prophase 1 is most extended phase and it can be further divided into 5 sub stages. We will discuss about that. Okay. So first of all, let's talk about the sub stages of prophase 1, and the very sub stage is what we call leptoteam or thin thread stage. So as you can see in the diagram as well, the chromosome appear as a thin thread like structure, right? We often say it shows a bouquet arrangement. Now what is the meaning of bouquet arrangement? You see, when the chromosomes are going to attach by both ends to the nuclear membrane, then we say it is a bouquet arrangement. Okay, and here what we see, that nuclear membrane and nucleolus are intact. Why they are intact? Because you see, disappearance of nuclear membrane and nucleolus is a part of a late prophase, and leptoteam is not a late prophase, it is the early prophase. Okay, so that's why it is there, right? So in leptoteam, we will concentrate only up till this point, that the chromomere are visible, we can say, and the chromosome appear as a long thin thread structure. The next comes the jagoteam, and jagoteam is called as a pairing stage. You know, prophase is also known for the condensation, or rather it is known for condensation. So in case of leptoteam onwards, what we see, that chromosome has started condensing, and in the process of condensation, what happens, maternal and paternal chromosome come close to it. In the process of pairing, what happens, maternal and paternal chromosome come close to each other, right? And the process is actually called as a synapsis. Okay, so homologous chromosomes get paired. This paternal and maternal chromosomes are homologous chromosome, and the pairing is actually, as I told you, is going to call as a synapsis or syndesis. Now there is a one more important thing that facilitate the very process of pairing, and that is synaptominal complex. Okay, so synaptominal complex is primarily made up of protein and DNA. So they actually help in bringing the like locus together or homologous pairing. So as a result, bivalents are going to be formed. So at the end of the jagoteam, what we can say, that bivalents are formed. Now often questions are asked. For instance, suppose if I say that 2n is equal to 10, then what is the total number of bivalents can be formed? The answer is 5, because if 2n is equal to 10, what does it means? The 5 paternal and 5 maternal is there, and both paternal and maternal together is going to constitute one bivalent. So haploid number is equivalent to the bivalents, right? Fine. Let's talk about the next stage, and the next stage is your packeting. Now packeting is actually called as a crossing over stage. That is the most important stage of the entire prophase 1. Here what happens, that the process of condensation is on. Since the condensation is on, what will happen? The chromosome will condense and they will split into chromatids. Okay, and when they will split into chromatids, what will happen? That tetrad is going to be formed, and once tetrad is formed, then crossing over is going to occur between the non-sister chromatids of paternal and maternal chromosome, and at the site of the crossing over, cross-sib structure is formed, what we call chiasma. Mind it, chiasma is not visible in packeting. Actually, it is visible in diploteam. That is the next stage. Okay. So this is the crossing over stage. and in order to make you understand, I would like to draw the diagram as well. So if I take, let it be, so this is a paternal and maternal chromosome, right, and this what we call bivalent. So let's take one bivalent chromosome. So let it be, it is a bivalent. So I am using two color just to make you understand. So this is the bivalent. Now as a chromosome keep on condensing, so it will splits into chromatids like this. So now it is splitting into chromatids. So again, paternal and maternal chromosomes are in the form of chromatids only. So what will happen, the crossing over is going to occur between these two structures. So we can draw like this. So that is the point where crossing over is happening, and this is what we call chiasma, right? So this is where the crossing over, and this is all about the packaging. Okay. Now let's discuss the next stage. The next stage is what we call diploteam. Now diploteam is what? They are moving apart stage. And why we are calling them moving apart stage? Because what happens that the paternal and maternal chromosome are start moving from each other. And why it is so? Because the synaptominal complex that was present in between them disorganizes. So if they disorganizes, what will happen? That like pole are going to repel each other. Okay, but they are connected at the crossing over or packeting. Okay, so what will happen? This crossover structure will start moving towards the terminal end. Okay, so suppose there's two chromosomes, these two chromosomes are moving away from each other. What will happen? This cross-shaped structure will start moving towards the terminal end. So the movement of the cross-shaped structure towards the terminal end, what we call terminalization. So this is the initiation for the process of terminalization. Now earlier also I told you that chiasma is visible over here. Okay. Finally, let's talk about the another stage, what we call dikinesis. And dikinesis is nothing but it is a late prophase. So all the character of late prophase are there. That means spindle fibers are going to be formed, nucleus and nucleolus is going to disorganize, right? And the chromosomes are going to be arranged in a random manner, as you can see there, and the chiasma stand fully terminalized. So you can see there that the chiasma are only at the terminal end. At the terminal end only they are attached. This is what we call chiasma are fully terminalized, right? Now let's see what is going to happen in case of metaphase 1. Now we know very well in case of metaphase 1 everything is going to occur at the center. So the fully terminalized structure, all those structures are aligned themselves at the center. Okay. So here the chromosome organize, that is the only things. Okay. Now let's talk about the anaphase 1. In case of anaphase 1, what will happen? That there is disjunction of the chromosome is going to happen. What is the meaning of disjunction? They are attached like this. Okay. Now they will separate. So this separation is actually called as a disjunction of the chromosome. Okay. So since disjunction is going to happen, so that's why it is the stage where actual reduction in the chromosome number happening. Okay. So the chromosome starts moving to the two respective poles, because here this is followed by the anaphasic movement also. So once the chromosomes start moving towards the two poles, the reduction in the chromosome number is going to happen, right? Now the telophase, in case of telophase, what we see that the two nuclei is going to be formed and both is going to be having a haploid one. Okay. Now let's talk about the phase between meiosis 1 and meiosis 2, what we call interkinesis. Okay. It is absolutely similar to interphase, but the only difference is that here they don't have a X phase. That means DNA is not synthesized. That is the only thing what you have to keep in mind, right?